Welcome to MLSQR. This is a follow-up question and answer session to our earlier tutorial on RBC indices. The following questions and answers cover the concepts, but are not exactly like those worded in our ASCP study guide. Spherocytes have a high MCHC in the CBC, and if you look in the peripheral blood smear, you'll see these round, small cells about 5 microns in diameter without a central pallor. This question is a little tricky because it requires our knowledge of the MCV equation and what is calculated and what is measured. By rearranging the equation following algebraic rule, we see that hematocrit is calculated from the RBC and MCV measurements. We remember from the last tutorial that RBC count and MCV are directly measured by the electronic pulse count and height, allowing hematocrit to be calculated. These findings are consistent with a microcytic hypochromic anemia such as iron deficiency, anemia of chronic inflammation, thalassemia, lead poisoning, and sideroblastic anemia. This question asks for interpretation and troubleshooting of a high MCHC that is suspicious for lipemia, an ecteric specimen, cold agglutinins, or instrument error. We could check for a clot. A common procedure is to incubate the CBC specimen for 30 minutes at 37 degrees to see if it corrects. If it does, this is consistent with cold agglutinins. We could spin the CBC and check for lipemia or if it is ecteric, or check the centrifuge chemistry specimen. We note the hemoglobin, MCH, and especially the MCHC are markedly elevated. Lipemic specimens are high in chylomicrons, which are rich in triglycerides. Icteric specimens are high in bilirubin. Both may affect hemoglobin measurements with the interference in the colorimetric analysis of hemoglobin using the cyanomet hemoglobin methodology. If you spin the specimen and see lipemia, you can perform saline replacement. We take out a precise amount of lipemic plasma and replace it with the same amount with normal saline. Mix well, and you're ready to retest for the CBC. And afterwards, verify the CBC parameters and RBC indices to see that if it corrects. Here's a bonus question not found in the ACP study guide that will test your skills in troubleshooting with your knowledge of the RBC indices. These CBC results show a classic pattern seen in hematology with a very low RBC count and H&H ratio less than 3 with a very high MCV, MCHC, and MCH. In fact, the H&H ratio is 1 to 2 instead of 1 to 3. What is your interpretation and your next step? Strong cold agglutinins, usually of the IgM type of antibody, will agglutinate several red blood cells to be counted as one giant cell, spuriously affecting the measurements and indices. The solution is to correct for cold agglutinins is to perform warm saline replacement or dilution according to your clinical lab's protocol.